what I want to do today with you is share some of our views on how cancer arises and some of the procedures that we can use to both manage and prevent the disease. Okay, I don't have any financial disclosures. Okay, I usually start my lectures, as some of you know. I like to look at where we are and where we need to go. And this is a slide that shows the cancer statistics from the American Cancer Society from the year 1990 to 2016. And it breaks it down into the new cases per year, deaths per year, and deaths per day. So I just simply take the number of patients that you see for yearly death, and you divide it by 365, and you get that number. It's just a little bit more like, what's going on every day. People say, well, there's good news and bad news in these numbers. The good news is at least from 90 to 2016, the rate of increase for deaths per year was slower than the rate of increase for new cases per year. So indicative of the fact that we might, in fact, be making headway in the movement against cancer. And that had to do a lot with the new diagnostics that were developed over those years, diagnosing more and more people with cancer. The numbers are going up in new cases, but the deaths per year were not going up nearly as fast, so that people think we're making headway. But then when you look at the numbers from 2012, the increase in deaths per year from 2012 to 2016, the percentage increase is actually higher than the percentage increase of new cases. This is an indication that we are not moving forward with this disease. So then the question becomes, we hear about it, the new breakthroughs. We hear about all these things on the news, the TV, making everybody so like, wow, look at all the great, wonderful things you're doing, look at all this and that. And I have to be honest with you, we have made some advances in some of the childhood leukemias and things like this. But, and then we're spending so much money. This is like, what is going on? The National Cancer Institute, how much money are they spending on cancer? Every 5 or 7 years we have this new big thing. Now we have the moonshot. This is another big thing coming down. And then we have the pharmaceutical industry spending billions and billions of dollars. Billions. We're running, jogging, swimming for cancer. Everybody's cancer, this, cancer, that. Where the hell is all that money going? What are they doing? Oh, we're doing research. What kind of research? I mean, can you imagine if we spent, it's like we got to the moon and back easier than we're doing this cancer thing. So I'm saying to myself, what is going on here? I use this provocative question only because Harold Varmus threw it all out in the NCI. Let's focus on provocative questions for the next 100 years. The issue here is, this is the fundamental issue in my mind. Is it a genetic disease or is it a mitochondrial metabolic disease? Most people don't even know about a mitochondrial metabolic disease. But we draw a little cell here. This is a simplistic thing. And I took away all the other markers for organelles. We have the mitochondria and then we have the nucleus. And the nucleus is the focus center of our attention for the disease, basically. And then we have this little bean-shaped organelle, and I think Dr. Kobayashi yesterday, it's much more complicated than this. It's like a spaghetti network around the whole inside of the cytoplasm of the cell. And, you look at this, and we have these two organelles. And these came from a symbiotic relationship very early in evolution, when these two organisms got together. One organism that knew how to capture the value of oxygen as an acceptor of electrons to make water, and the other one was fermenting like crazy. But these two guys got together in a symbiotic relationship and created a more complicated structure. And this structure eventually led to us and more complicated, more complex organisms. And there is a remarkable discussion that goes on in every one of our cells, the discussion between that little energy-generating organelle called the mitochondria and the nucleus. And that's called retrograde signaling. So there's a constant communication in every one of our cells, and that goes throughout the whole body, because these mitochondria are linked generally throughout the body. There's slight differences between liver and brain and heart and things like this, but they're basically the same organelle. And they communicate with the nucleus through a very sophisticated molecular signaling pathway. And when things in the homeostatic environment of the body change, the mitochondria will signal the nucleus to change its gene expression in response to whatever the stress happens to be. And we've heard a lot about these kinds of things, but I'm just giving you an extremely simplistic overview of what's going on inside the cell. So the question is, we have a disease, and we know we have genetic abnormalities, and the nucleus becomes, as they said, shot to hell. That was Harold Varmus's, Bert Vogelstein's discussion. You know, the genome is shot to hell in cancer. But the mitochondria are also damaged as well. And now we have to focus on, you know, what's cause and what's effect and what's going on here. So I use this term, the dogma. The dogma right now is cancer is a genetic disease, no question about it. And this was solidified in the Hallmarks of Cancer by Drs. Hanahan and Weinberg. Cancer cells carry the oncogenic and tumor suppressor mutations that define cancer as a genetic disease. Now, when I use the term dogma is a very powerful term. Okay, it's a belief system. It's an irrefutable truth. It's not to be questioned. It is now, this is the way it is. It's a dogma. It leads to indoctrination. We don't discuss this anymore. And I know this because I teach these classes. I teach general biology, I teach genetics, I teach biochemistry. And if you go to the section in the book, the textbook on cancer, cancer is a genetic disease. Go to the NCI website, look it up. What is cancer? Cancer is a genetic disease. We don't talk about anything else. It's just a genetic disease. So what this has done now, over the last 50 years, is all these textbooks, so we get the students in biology in the freshman class, even in high school, advanced, placement classes, cancer is a genetic disease. You go to medical school, cancer is a genetic disease, right through the whole educational system. So we've essentially indoctrinated generations of scientists and physicians to think that cancer is a genetic disease. It comes on the news at night. Sequence your genome to help your cancer. So there's no discussion about this, anything other than that. So the somatic mutation theory is the foundation upon which the gene theory is based. Mutations in genes inhibit and stimulate cell division and contribute to cancer. So they use these little diagrams like an automobile that's speeding out of control, that has no brakes, to indicate what's going on inside of a population of tumor cells. They're growing out of control and they can't stop growing. And this is due to mutations in oncogenes and tumor suppressor genes. And this is the way they convey. And of course, as you go through the scientific educational process, each new level of education goes deeper and deeper into the signaling cascades that we heard about earlier today from Dr. LaValle. And the students have to memorize all of these signaling cascades, just like I think Dr. LaValle says, you don't memorize all the streets in Dallas, but we force our students to memorize all those signaling cascades. Torture, they hate it. I can tell you that. So, and then we've developed this view of cancer as a group of hallmark events, that involve stimulating proliferation, evading suppressors, growth suppressors, activating invasion, enabling replicative immortality, including inducing angiogenesis or vascularization, resisting cell death. So each one of these so-called hallmarks of the disease are the subject of an enormous research effort and an enormous pouring of research dollars into trying to figure out every one of these major hallmarks of the disease. And now, with their latest paper, the new, that was 2000. In 2011, they came out with another rendition, an update of what was going on, even more levels of complexity and obscurity. If you ever read the original paper like we do in my cancer class, we go through the entire paper, the original and this one. We have now emerging hallmarks, dysregulated cell energetics, avoiding immune destruction, and then some enabling characteristics like, tumor promoting inflammation, genomic instability. But if you read the paper very, very carefully, in the quotes that they have in the paper, because I'll be focusing on the metabolism, the dysregulated cell energetics is now considered an emerging hallmark that is programmed by proliferating inducing oncogenes. You see that? By its programmed, by differentiation. This is preserving the dogma. Preserves the dogma. It is a genetic disease, and the genes are controlling the abnormal metabolism, and this is what I'm going to challenge. But we have to understand we need to understand, before you can challenge a field, you better darn well make sure that you understand, what the nature of this whole thing is. As it was pointed out, I got my degree in genetics from a master's degree from Illinois State University in classical genetics and a PhD from the University of Illinois in biochemical genetics. So I know genetics. It's not like it's a foreign field to me at all. So now we look at this accumulation of random mutations as the development of cancer leads to the epithelial mesenchymal transition. This is where we take an epithelial cell. So it's really not an epithelial cell in this simplistic diagram, the one on the left. As you go further to the right, you get this little dysmorphic amoeboid-looking cell, and that's mesenchymal phenotype. It's no longer linked or associated with other cells. It's kind of like, and that's called the mesenchymal phenotype. So we have a series of random mutations that are going to convert an epithelial cell into this malignant cell that has a mesenchymal phenotype. How many mutations are responsible for this transition of a normal cell into a mesenchymal cell? Nobody really knows. There's 4, 5, 6. There may be 1 million of these mutations, according to Michael Stratton, Sir Michael Stratton from the UK. So we're really not sure, but this is the way they think it happens. And then, of course, the end result of all this is individual cancer cells are genetically tested for personalized therapy. And precision medicine, personalized therapy, this woman staring into the screen looking at some blobs or iridescent dots, and then she's going to tell you or tell someone how we're going to use that information to better manage your disease or do something that's going to be related to the molecular problem that you have. And we hear this advertised on the news. Right? Cancer Treatment Centers of America. We're going to personalize your genetic profile to determine how we're going to be able to treat your cancer. Then Dana-Farber in my neighborhood comes out and says, no, we can do it better. We're going to do all the gene screens. And D. Anderson comes on. What's going on? Everybody's advertising now for personal medicine to take your tumor and look at all the mutations, and we're going to make a decision on your health based on this kind of information. And everybody says, it must be important. So the real important issue here, of course, is metastasis. This is what ultimately, either metastasis will lead to the demise of the individual, or the attempts to manage metastasis will lead to the demise of the individual. Okay, what is this? It's a very stereotypical cascade of events. It's remarkable. Whether you have lung cancer, bladder cancer, breast cancer, colon cancer, when those cells begin to spread out of the primary site, they go through a stereotypical cascade. They have a local invasion. You can see in this image here, the green cells are the tumor cells. So you can see them starting to break through the basement membrane, invading into a local area. Intravasation, that means they leave their local site, enter into the bloodstream. And this is a very sophisticated sequence of biological events. You can't believe how many reactions are involved in that process. Then they get into the bloodstream, and they evade the immune system, and they also suppress the immune system. And I'll talk to you why the metastatic cell is actually a macrophage. It is part of our own immune system, and this can explain a lot of the phenomena that we have, and I'll talk about that a little bit later. And then they extravasate at a distant location. So they go through the bloodstream, and then they come out of the bloodstream, and they make these secondary tumor sites somewhere else, secondary tumor formation. My question that I raise is, how is this stereotypical events, this cascade, how does this happen from a bunch of random mutations? It doesn't make any sense. How can we get, how can randomness produce non-randomness, according to the gene theory? So I use this, this slide, it's on social media now, it's all over the place. You can see it everywhere. I don't know, I put it somewhere, I can't remember where, but here it is. How do we advance new therapies for cancer management and prevention if this 800-pound gorilla thinks cancer is a genetic disease? So the question is, who's the 800-pound gorilla, right? It's the National Cancer Institute. It's the American, the academic and pharmaceutical cancer industries. These are powerful organizations, extremely powerful organizations. They have locked in the dogma. The dogma that cancer is a genetic disease permeates almost everything that we do, and it's sanctioned by the federal government. Evidence that challenges the dogma. And when I talk, that's what I said. I write these papers, I don't hear anything back. Nobody's attacking me. He's wrong. He said that. They don't want to look at it. They don't want to talk about it. They don't want to hear about it. It's called convenient selective deafness or blindness or ignorance or whatever you want to call it. All right, so let's look at some data that I like to show. Now, I'm only giving you a very small snapshot of the real issues. Okay, this was done by Dr. McKinnell's group, published in Science, top scientific journal back in '69. This frog has a tumor, kidney tumor, kills the frog very quickly. This strain of frog gets these kidney tumors. You can see it there with the arrow. Okay, so what McKinnell did is he took the cell out of the tumor. You can see it down here on the bottom. The cell, you can take the whole cell out, and then you could surgically remove the nucleus of the tumor cell and take that nucleus by itself containing the oncogenes and the tumor suppressor genes, and they put it into a new cytoplasm from a fertilized egg, but they took the nucleus of the egg out, so you have all the normal cytoplasm, but you put the tumor nucleus into the egg, and you got that picture of the tadpole there, and everything was normal. All right, the tadpole looked great. They went through the tadpole, the 100 different slide sections. They couldn't find a single dysmorphic growth of cells in there. The only problem was the tadpole could not mature into an adult frog. So there was something in the tumor nucleus that blocked the development of the frog, but it didn't cause cancer. Now, so what did they do? Nobody said anything about it. It's an anomaly. Doesn't support the hypothesis. Beatrice Mintz and Carl Amensi from Thomas Jefferson in Philadelphia, leaders in the field of developmental biology, they took a malignant teratoma growing out of control, and they took the whole cell and put it into a blastocyst of a whole bunch of mice, 230 mice. And all the mice developed normally. They didn't have any cancer. And then the offspring of the mice produced offspring that didn't have any cancer, yet they were all generated from the cell of a malignant teratoma. What happened? What they said, conversion to neoplasia does not involve structural changes in the nuclear genome. What does that mean? You know? So it's not consistent with the dogma. Forget about it. We don't talk about those things. Then we have another paper from Jim Morgan's group at St. Jude's. And what they did was they took a medulloblastoma, cerebellar tumor, and took the nucleus out of the cerebellar tumor and put it into an embryo, a stem cell, and they cloned the mouse from the nucleus of the medulloblastoma. And there's the picture of the mouse with the normal arrangement of ectoderm, mesoderm, and endoderm. And they said, although medulloblastoma-derived embryos aborted, just like the frog couldn't develop, the mouse, they aborted, none exhibited uncontrolled proliferation resembling tumorigenesis. The findings are inconsistent. You'd have expected some dysmorphic growth. They contain the tumor suppressor mutations and the oncogenes. That's what's driving cancer, according to the dogma. We're not seeing it. Anomaly. And then Rudy Anish and his group at MIT cloned mice from the nuclei of melanoma. And in this particular study, they took the melanoma, malignant skin tumor cells, tumors, and characterized all the mutations in this malignant melanoma, and then took another nucleus and put it into an embryonic stem cell, and there's the mouse cloned from the nucleus of the melanoma. The presence of major genetic abnormalities in embryonic mice cloned from the tumor nuclei provides unequivocal genomic evidence that the mice were cloned from the tumor nucleus. But they didn't have cancer. They didn't do the things that we're supposed to be doing. So they say reprogram. What does that mean, they reprogram? You got to mutate. You supposedly have all these somatic mutations that cause the disease. How are you going to reprogram a whole bunch of mutations? Doesn't make any sense. Inconsistent with the dogma. Then Benny and his group from Baylor College of Medicine, he did something different. He transplanted mitochondria from one cell to another. So he had these very aggressive breast cancer cells that were highly metastatic. He removed the mitochondria now, And then he put new mitochondria in from a normal cell and completely shut down the malignant properties of these breast cancer and turned off all these oncogenes from the transplanting of the mitochondria. And then he did the reverse. He took the mitochondria from the most aggressive breast cancer cells and moved them to a very slow-growing tumor and made the tumor grow wild. The mitochondria were calling the shots. So I published this paper. It was based on chapter 13 of my book, and I just updated all the references, and I even put even more stuff than what I had in my book. And I brought it all together, and I said, okay, guys, you choose. Here, we're going to do a compare and contrast. We have the dogma, and we have the mitochondrial metabolic theory of cancer. Why don't you just read the evidence, and you make your own decision about this, okay? So here's all the evidence. Here's all the original research articles. Here's all the talk of Hanahan and Weinberg and what's going on, and here's this information. How do you incorporate it? And what I just showed you was only a snapshot. You should see all the other papers that are pointing in the same direction as the information that I just gave you. What's going on here? Okay? There's a very compelling argument that cancer cannot be a nuclear genetic disease. So this is another image that one of my students drew for us to put in the book. And this gives you a summary of what I just said. Green, normal cells beget normal cells. The red cells, tumor cells beget tumor cells. But inside that tumor, we have damage to both the nucleus and the mitochondria. They're both damaged in this cell. So what's causing this? According to the dogma, it's the tumor suppressor genes and the oncogenes in the nucleus that are driving the dysregulated cell growth. But when you take the tumor nucleus and now put it into a new cytoplasm, you don't get that phenotype. In fact, you can see we had mice that were cloned, frogs and things like this. But when you do the opposite, you take the nucleus of the normal cell and you put it into the cytoplasm of the tumor cell, you either get dead cells or you get tumor cells. You don't get normal cells. So what these arguments, these data do not support this whole concept that genes of the nucleus are driving the disease. You'd get just the opposite results, not what these results are showing. Just the opposite. This is devastating news to the gene theory. If the gene theory is not correct, what kind of disease is this? Think about it for a minute. So if somatic mutations are not the how do these cancer cells arise if genes aren't involved? That's the key. Genes are... Okay, Warburg had it pegged a long time ago. Cancer arises from damage to the respiration. Energy through fermentation gradually compensates for the respiration. Cancer cells continue to ferment lactate, and we've extended this now. You know, I filled in the blanks from what Warburg was unable to do at that time, because we can have amino acid fermentation, which is an alternative method of getting energy without oxidative phosphorylation. Enhanced fermentation is the signature metabolic malady of all cancer cells. That's a very important point. So, if we look at a tumor, and this tumor has all these different kinds of cells, and we've heard so much about this. Yet, when every cell in that tumor is a different genetic entity, you do a gene screen, you pull out all the cells, and you look, everybody has different kinds of mutations. No two tumors have the same mutations. No cells within the tumor have the same mutations. But all the cells are fermenters. They're all fermenting in one way or another. They're either fermenting glucose or an amino acid. They're fermenters. So the question we have to ask ourselves, do you think it might be better in our approach to managing this disease if we focus on the metabolic malady common to every cell in the tumor? Or do you think we should look and try to kill the tumor by looking at every single different kind of gene mutation that's different in every cell in the tumor? Apparently, it's the more complicated way. So when we look at energy, and this is again from one of the general biology books, you know, most of our energy, and we heard that from Dr. Kobayashi, most of the energy comes from our breath. We breathe in, we exhale. The waste products are CO2 and water. And we get most of our energy from this organelle called the mitochondria. Now we see that we have a little energy from glucose in the cytoplasm through the glycolytic pathway, and we have a little bit of energy in the succinyl-CoA synthase step in the Krebs cycle. But by and large, it's mostly respiration. The cancer cell is different. The cancer cell is generating a significant more energy from these primitive substrate-level phosphorylations and much less energy from phosphorylation, and that's coming from the damage to the respiration of the mitochondria. So the cancer cell, as Warburg said, if you can't breathe, you're going to ferment. And if you can't ferment, you're going to die. So when we look at the structure at the electron microscopic level of mitochondria and tumor cells, we see rather dramatic changes. And I've looked at every kind of a cancer where we can find EM, electron microscopy. And this is just one example, a glioblastoma multiforme mitochondria compared with a normal mitochondria. And the normal mitochondria has those beautiful little stripes. And again, this is like a tube, so you really cut, this is like a spaghetti, and you just cut one section of it. But those little stripes contain the proteins and the lipids of the electron that drive the electron transport chain and is responsible for our generation of energy metabolism. So you look now at the mitochondria of the tumor cell, and you can see its crystallysis, the breakdown of the cristae. So you have ghost mitochondria, and you have fewer mitochondria, and there's all kinds of abnormalities in these tumor cells in the structure and function and number of mitochondria. How is this cell going to generate energy normally if the very organelle that's responsible for this is in some way defective? It's not. So if that cell is going to stay alive, it has to ferment. And that's what all these cancer cells are doing. They're fermenting. The fastest growing cells of cancer ferment the most, and the slowest growing cells of the cancer ferment the least, but they all have some fermentation. So if you're going to change the view of what cancer is, you just simply can't discard decades and decades of research that have already been done. You have to just reinterpret the